Hello, everyone. Good morning. Welcome to the last day of the second symposium, the biology of galls. Let me introduce myself first. And again, my name is Gabriel Barros da Silva. I am an undergraduate student of biological sciences at the State University of Feira de Santana in Brazil. I am a member of the chapter Iniciação Botânica, a group of young botanists affiliated to the Brazilian Botanical Society, and 4 Brazilian universities from 3 Brazilian states are involved in this project, whose aim is to introduce biology students in botany subjects and promote and support scientific events. This is the first event that the chapter is hosting this year, and I would like to invite all of you to follow us on social media, Instagram, because we have exciting news and events upcoming. All right. This is the last day, and we'll have group 5. It's called Gall structure and functioning. We will have 3 students show, actually 3 researchers to show the research. The first one will be Ravena Malheiros Nogueira, a PhD student in plant biology at the Federal University of Minas Gerais, Belo Horizonte, Brazil. Mariana de Souza Costa Freitas, master student in plant biology at the Federal University of Minas Gerais, Belo Horizonte, Brazil. And Igor Abba Arriola, PhD student in plant biology at the Federal University of Minas Gerais, Belo Horizonte, Brazil. Let me put them on the screen just to say hello to everyone.
Good morning.
We put Mariana on the screen. All right. I'll show the video poster right now, and then we will have the round of questions, and I'll put all of you on the screen again. All right. Let me share my screen now. The first one will be Ravena's video. Let's wait.
Hi, I am Ravena, a doctoral student in the
full screen. Okay.
postgraduate program in plant biology and UFMG.
Are
I
y'all
am an
listening
institute
well?
Professor José Isaías, and now I will present the research entitled Several Traits of Galls in Caatinga Environments by me, by Helena Cotrim Costa, and José Isaías.
The Caatinga is characterized by abundant solar energy, water deficits, low relative humidity, and irregular rainy season. The Caatinga environment to impose constraints on the development of houseplants and in their associated galls. Mosatherium flora is a common species in semi-arid environments which hosts a bivalve-shaped leaf gall induced by Lopez mosaic.
Changes in cellular structure occur along developed galls and in response to water stress. We used samples of non-galled pinnahacs and mature galls.
The samples were subject to anatomical and immunohistochemical analysis to map the traits that can be expressed in Caatinga environments. We used monoclonal antibodies, LM20, LM5, LM6 for the detection of vaccine epitopes. The pinnahacs present inside epidermis, isogeometric paradigmatic cells, and bicollateral vascular bundle. The mature galls have uniseriate epidermis, common tissue, scalenniculum layer, and collateral vascular bundle form the gall outer compartment, while the typical nutritive tissue forms the gall inner compartment.
In pinnahax, the epitopes of mechistratified homogalactic neurons were marked by elegant joints on the parenchymal cell walls. The epitopes of arabinans in cell walls, epidermis, and the cell walls of the adaxial cortical parenchyma and of the pinnahax floor. In Gauls, the epitopes of mature stratified homogalactic neurons were marked on the cell walls of the common tissue, the vascular bundle, and the typical nutritive tissue. The epitopes of galactans were marked in the cell walls of the Gaul common tissue, and arabinans were marked in the cell walls of epidermis, common tissue, and typical nutritive tissue. Homogalactronans, in association with arabinans and galactans, provide greater porosity to the cell walls and are key elements for cellular hydration. The galactans and arabinans confer flexibility and hydrophilic traits. The dynamic of pectins favors the development of myostatin flora without shaped galls and adverse environment conditions of decaying. Thank you very much for your attention.
Hello, I am Mariana Freitas and I will present the work entitled Wool Apple Aphid Stem Galls. The wool apple aphid, Eriosoma laniger, is a pest crop that attacks the root and stem of apple trees worldwide. Even though Eriosoma laniger is an exophytic aphid, its feeding on the phloem results in galls, affects the water conductivity of the host organs, and reduces the tree vigor. The Eriosoma produces a wax covering that protects its body from environmental factors. The aphid stay on stem and root barks, and despite no larval chamber is formed, there are peculiar tissue alterations. The host species, Malus domestica, has worldwide economic importance, being the third fruit production of the world. Thus, the wool apple aphid represents a great economic risk for apple producers. The study of Eriosoma lanigerans gall development on Malus domestica aims to describe the anatomical changes caused by the aphid and find windows of opportunity to block the infestation. Non-galled stems have a compacted tuberized periderm with lenticels. The cortical parenchyma has phloem fibers arranged in arcs around the vascular system, and the cambio produces a continuous cylinder of secondary phloem outward and secondary xylem inward. The stem galls are fusiform and recognized by the intumescence of the galled stem as a product of intense cell division in the xylem region, stimulated by the sucking activity of Eriosoma laniger, and the cambio and phloem occupy the periphery of the organ. The feeding activity stimulates the cambio to overproduce parenchymatic cells inward, a few but wider vessels element, and generates a cell reorientation both in radial and axial system. The main aphid feeding site is the phloem, but the major change in the secondary xylem suggests that the aphid also feeds in water-conducting cells. The observed anatomical traits could confirm that the feeding site of the Eriosoma goes from the phloem up to the xylem cells, since this aphid could insert its stellate until the xylem, as seen by Sandanayaka and Hale in 2003. Next steps will look for evidencing the energetical molecules related to the nutritional requirements of the aphid and for checking its effect over the water conductivity. With the information obtained through anatomical analysis and the performance of subsequent tests mentioned before, it is possible to determine sites to block the aphid's feeding, consequently reducing the infestation. This is all for the moment. Thank you for the attention.
Hello, my name is Igor Arriola, and I'm a PhD candidate in plant biology at the Federal University of Minas Gerais. I'm a researcher associated with the Neotropical Gall group, focusing on Gall anatomy and metal accumulation in galls. Today, I will present the work entitled Nematode-Induced Galls Show Host-Specific Signatures in Their Structure, which is a synthesis of a work published by me and Dr. Jose Isaias. Well, unlike the root node nematodes, Digitilex galliformans is a plant parasitic nematode that induces galls on aerial organs of several species in the genus Myconia in the Brazilian Cerrado. These galls were previously described as occurring on leaves and inflorescence of Myconia albicans and Myconia ibaguensis. And as an outstanding feature, these galls have an indeterminate growth capacity. This peculiar feature is conferred by the totipotent capacity of the nutritive cells that outline the chambers with the nematodes. In this work, we described the structural profile of the galls induced by Digitilex galliformans on leaves of Myconia corallina and Myconia lacunosa, and compared these galls to those previously described on Myconia albicans and Myconia ibaguensis by cluster and principal component analysis. As we expected, the galls induced on the four host plants presented several structural similarities, including the indeterminate growth potential. Unlike most gall insects that are monophagous, these nematodes are also specialists, but they are oligophagous inducers with a strict relationship with Myconia. However, some traits such as the trichomes and emergence diversity expressed in the galls were determined by the host plant constraints. These traits of each host plant species supported separation between Myconia lacunosa and the PCA, and the clustering of the other three Myconia species, which looks like to coincide with the phylogeny of the Melastomataceae family. For more details, please check the full article published on Flora. I want to thank the organizing committee and the funding agencies. Have a good symposium and see you. Bye.
All right, let's go to a round of questions. I would like to congratulate all of you for the research, and I'll display the questions on the screen. I would like to congratulate all of you for the research. Okay, the first one is to Ravena's research. Bruno Garcia Ferreira asks, Ravena, do you believe that the responses in cell walls of galls are related to the adaptations of the plant to Caatinga environment? We can hear you. It's your microphone is off. Sorry.
Sorry. Maybe is, maybe not. They maybe a plant characters that favor success in Caatinga environment.
Okay, you done? Next one is for Mariana. Bruno Garcia Ferreira asks, Mariana, these galls are induced in secondary stems, trunks, or young ones?
Well, these galls are induced in the entire tree. So it is induced in secondary stems and young ones, but mainly in the young ones.
All right. Next question, directed to Ravenna. Graciele Bragança asks, Ravenna, congratulations for your presentation. In your work, did you also analyze something related to hydraulic conductivity?
Well, I did not analyze it yet, but it is in my future plans.
All right. To you again, Ravenna. René Carneiro asks, have you been able to find similar profiles when compared to other galls? Which cell wall component is the most common or abundant?
There are differences when compared to other FABAs. The immunocytochemical analyses are qualitative and not quantitative. The strongest leveling with for homogalacturonons.
All right. The next one is directed to Igor. Bruno Garcia Ferreira asks, he congratulates you first, and then he asks, these analyze confirmed. What are you expecting?
Thank you for the question, Bruno. Yes, in the initial steps of this work, the plant known in this work as Myconia lacunosa was previously known as Leandra lacunosa. And in the conduction of this work, the Leandra lacunosa was included in the genus Myconia. So the separation between Myconia lacunosa and Myconia species is what we expect to find in this project.
René Carneiro asks, Igor, do you think the constraints imposed by the host plant also play an ecological role for the galls and nematodes?
I don't know, Rune. I think that the diversity of trichomes and emergence on the Myconia species is a convergent trait, so they are a redundant feature on these galls.
Another question to Ravenna, and this is the last one on our chat. Edgar Poli asks, Ravenna, the quantification of cell wall components in xylem and phloem, according to Rene's questions, can be of great importance to discuss your results. Sorry, this is not a question. This is just a comment, but yeah, sorry.
Yes, Professor Edgar, I count on you to help me.
All right, thank you, all of you. I'll ask now I'll put down the screen the second group of this morning. Thank you all again. See you soon. All right, now I'll invite, let me check their name first. For the second round of presentations, we will have Ana Flávia de Melo Silva, PhD student in plant biology at the Federal University of Minas Gerais, Belo Horizonte, Brazil. Pedro Henrique Pereira Gonçalves, PhD student in plant biology at the Federal University of Minas Gerais, Belo Horizonte, Brazil. And Graciele Pereira Pimenta Bragança, doctor in plant biology by Federal University of Minas Gerais. And now she has a postdoc position at the Federal University of Minas Gerais, Belo Horizonte, Brazil, with a fellowship by Reza FAPERJ. All right, let me put down the screen. Okay. All right, I'll show now your video posters. The first one will be Ana Flavia's video, all right? I think I didn't allow the audio. Just wait a second. I messed up. Sorry. Now we have the audio.
Hello everybody, I'm Ana Flávia. I'm a doctoral student at Federal University of Minas Gerais, and this work that I'll show you had a collaboration of Tiago Magalhães, Denis Oliveira, and Rosi Isaías. And the title is Anatomical and Histochemical Implications of Sexually Dimorphic Galls Induced by Eriagalococcus Isaías, Eriococcide, on Pseudobombox grandiflorum, malvaceae. Eriococcide is a family in the order Hemiptera. Several genera of Eriococcide have sexual dimorphism, which is reflected in the gall phenotype. The males and females of Eriagalococcus isaias induce galls on a decidual host plant, Pseudobombox grandiflora. The duration of life cycle of the males are shorter than that of the females. However, their galls are morphologically similar. So, in this work, we evaluate if the galls reflect the sexual dimorphism in an anatomical level and if the histochemical profiles of primary metabolites such as starch, proteins, reducing sugars, and lipids are distinct between the sexes of Eriagalococcus isaias on leaflets of Pseudobombox grandiflora. The galls are green, intralaminar, conical-shaped, and have a single central chamber. They are covered by uniseriate epidermis and have a homogeneous cortical parenchyma, which can be divided into two compartments, the inner compartment and the outer compartment. Vascular bundles are located near the nymphochamber, and in the outer compartment is possible to see many mucilaginous cells. The sexual dimorphism of the Eriagalococcus isaias results in subtle structural difference in the mature stage, as the shape of the nymphochamber. The galls induced by females have a bigger and rounder nymphochamber, which assumes the female body. The galls induced by males have an elongated chamber similar to the males' bodies. The cells of the top of the chamber have phenolic substances, with visually higher concentrations in the galls induced by females when compared to the galls induced by males. The primary metabolites are related to the maintenance of the gall structure. Starch grains were absent in galls induced by males, which does not feed during the pulp stage, and does not appear to stimulate a strong sink of photostimulates. However, galls induced by females store starch, which indicate a stronger sink due to metabolic cell demand regarding the longer life cycles of the female. Proteins and reducing sugars were detected in the inner tissue compartment, with accumulations decreasing toward the outer layers of the gall cortical parenchyma for both sexes. The reducing sugar available are energy sources that are rapidly assimilated to maintain the growth and development of the males and females gall tissues. Finally, lipid droplets were detected in the cells of the inner tissue compartment. and this detection was more intense in galls induced by females. The deposition of suberin in the cell walls at the top of the chamber act as a barrier against any biological or physical stress during the male and female galls development. So, this current data corroborate the hypothesis that the sexual dimorphism of Eriogallococcus isaias reflects in the structural and histochemical profiles of the galls. That's all. Thank you. Bye-bye.
Hello everyone, my name is Pedro Henrique and I'm a student on the Graduate Ecology program here at the Federal University of Uberlândia. My advisor is Dennis Coelho de Oliveira, and today I will show to you a little bit about research that I did entitled Could the sexual dimorphism of an gall-inducing insect determine different morphotypes of galls in a host plant? For you to first understand my work, I need you to understand what are galls. Galls are neo-formed structure by the chemical and or mechanical stimulus of some organisms. These organisms can be insects, nematodes, and archaea, and especially in insects, they has a higher range of inducing galls, and usually one species just induces one morphotypes of galls. But when one species of insect can induce different morphotypes of galls, these sometimes can be related to a sexual dimorphism of the gall-inducing insect. And this induction of different morphotypes can indirectly increases the plant resource variability that galling insect can assess. So here I brought you an example, Lopesia matype, that is a Diptera cecidolity. This insect is capable of inducing two different morphotypes in the leaflets of Matyba guianensis sapindaceae. So, by going to the field, we raise this question. Are these differences related to the sexual dimorphism, determining different morphotypes of galls? And to test that, we collected 124 galls, half from each morphotype, to test the hatching of the adult insects, the morphology, the anatomy, and the histochemical attributes. And with the results, we can see here from the 62 bigger morphotype put to emergence, to hatching, 36 females only hatched from this bigger morphotype. And from the 62 smaller morphotypes, only 13 males hatched only from this morphotype. And with this result, we can testify that the sexes occupied different gall morphotypes. And what about the morphometrics analysis of these galls? Here we can see that they are significantly different in height and width. And we can see here with this graphic that this is true. They are very different in height and width. So the females and males occupy different morphotypes. And when it comes to anatomic features of the galls, we can see a higher range of sclerification, usually below the larval chamber. And these galls, they are formed by the cell hypertrophy, the tissue hyperplasia of the spongy parenchyma. So the spongy parenchyma is what forms the gall here. And we also can see a higher range of vascular bundles in both morphotypes. But when it comes to histochemical features, there were no differences in histochemical compounds detected. They both have starch and reducing sugars as primary metabolites. So when it comes to morphometric analysis, when we see this picture, we can see the compartmentalization of tissues. We see two different tissues, one tissue with globoid cells and another with elongated cells. But the measurements were already made, and they are still in process of statistics and discussion. In the literature, the Cecidology males usually emerges earlier and have a shorter life cycle than the Cecidology females that stay longer in the galls and have a longer life cycle. So these differences of lifespan can be a possible explanation for the occurrence of these different morphotypes. So when we see that the Gall shape determination depends on the constant Galling feeding behavior, we can see that this behavior simulates differently the process of Gall tissue's development. So females, by staying longer in galls than males, they may promote more stimuli in the plant, which can result in the formation of different gall morphotypes for each sex. Here I want to thank you all for your attention, and I brought a little phrase that is very good to think about. Little strokes feel great oaks. Even though something may seem impossible, if you break it up into small parts and take one step at a time, you will succeed. That's it. Bye.
Hi, my name is Graciele, and I will talk about xylem differentiation controlled by Cecidology and Gangois Fabas. This work was done by me, Mariana Freitas, and Rosi Isaías. The data of this presentation is published in the journal Quiz. The galling herbivores influence the development and differentiation of plant tissues, forming new structures, the galls, with new patterns in dermal, ground, and vascular system. The new pattern of xylem differentiation is influenced by the action of phytohormones, auxins, and cytokinins, and the position of the gall in its host organ. The galls can be extralaminar when attached to the host leaf by a peduncle, or can be intralaminar when they're in a morphological continuum with the host organ. Our study model was in angoids, which host three gall morphotypes. Globoid and lenticular galls are extralaminar, and fusiform is intralaminar. We collected the material and analyzed of the pattern of vascular tissue differentiation, cytometry, and histometric features of xylem cells, and the detection of auxins and cytokinins. The detection of phytohormones is related to the differentiation of new vascular cells. In extralaminar galls, dioxin was detected in the outer tissue compartment. In this region, vascular bundles were also developed that connected with the vascular system of the host plant. The cytokinins were detected in the nutritive tissue and vascular bundles. The development of extralaminar galls theoretically imply in rupture with the host leaf ontogenetical pattern. This rupture and the continuous flow of auxins are associated with the origin of the vascular tissues, parenchymatic cells, and the differentiation of narrow vessel elements. Intralaminar galls, dioxin was detected in the outer tissue compartment and cambion zone, and cytokinins were detected only in the cambion zone. Intralaminar galls, the cytokinins increasing the sensitivity of auxins in the vascular cambion and associated to continue with the host organ, implying the differentiation of wide vessel elements. The xylem morphogenesis involves the synergies between auxin and cytokinins in cells recruited to redifferentiate in vascular elements, both in the extralaminar and intralaminar galls on gangoids, to ensure efficient hydraulic conductivity. Three strategies are triggered. One, a high rate of differentiation of parenchyma cells in extralaminar galls. a high differentiation of vessel elements in the intralaminar galls, and 3, difference in the width of vessel elements in both gall morphotypes. Thanks.
All right, congratulations, all of you, for your research. We have some questions on the chat. I'll put them on the screen. The first ones are directed to Ana. René Carneiro asks, Ana, in terms of tissue stimulation, what do you think males and females do differently inside the galls to determine such anatomical and histochemical difference?
Well, René, thank you for the question. The females stay for more time inside the galls, and they feed during the second instar, while in the adult stage and gets pregnant, while the male they only feeds on the galls only the first instar, and then he doesn't fit more. So I think these features are the how can I say, I'm sure it's the difference that determines such anatomical and histochemical differences.
All right. Bruno Garcia Ferreira. Anna, nice work in the images. Do you believe that cell metabolism, ultrastructure, or cell wall composition may also be distinct in these sexually dimorphic galls?
Bruno, yes, maybe it is. In the future, we are do this test, so I can be answer you later.
Lubia Maria Guedes Garcia asks, Anna, good morning. Do females and males feed on the same gall site?
Good morning, Lubia. I don't know if the females and males feeds on different sides on the gall at different places, but also both feeds on the vascular bundles. I don't know if there are different sides.
Okay, Bruno Garcia Ferreira asks to Pedro, you are very didactic. Congratulations. How did you discover that these galls were induced by the same species? And do you have any hypothesis to explain the variation in these galls?
Thanks, Bruno. It is an honor to have a compliment like that. So when I was going to the field with Oyara, that and at the time was my co-advisor, she started to plant the seed in my mind that these galls could be from the same species. And by hearing the galls in laboratory, we could see that, and I forgot to put this. I didn't forget. It's just that I hadn't time to put this in the presentation. But when we opened these galls in laboratory, we could see that there were all the instars of the insect, larvae, and pupae in same morphotypes. And how is it possible to have pupae morphotype on a gall that is smaller than the bigger one? So we discovered that these galls were phases of development by seeing the pupae of the larva in the smaller morphotype. And by that, we raised some hypotheses that this could happen because of the lifespan and the life differences of the sexes of this gall-inducing insect.
All right. René Carneiro. He's doing the same question he just asked to Anna. What do females and males do differently inside the galls to determine the changes that is only due to lifespan inside the galls?
I think that might be because from what I've tested, the primary metabolites tests that I did, the histochemical tests, they didn't have any differences in compounds in these galls. And also the structure are very similar. They mostly differ in size, thickness, the height and width of the galls. And we are still analyzing some data from the stometric and cytometric measurements that I made, but I think that might be due to the lifespan. But it is hard to test that, because how can I see that the male and female have differences in, I don't know, days of living? It is a very hard subject to understand.
The next question is by Jack Schultz, and he asks to Anna and Pedro. Are there any difference in the location of galls initiated by the two sexes? Do they exploit different cell and tissue types? Both of you can answer this. Anna,
in my case,
I thought your microphone was off.
In my case, both female and males insects induces galls on the abaxial leaves. There are no difference in the locations. If they exploit different cells or tissue types, no, they can't.
In my study case, both of the sexes, they occupy galls on the abaxial surface of the leaves, but we can also see some galls in the adaxial portion of the leaf. But when it comes to the exploit of different cells or tissue types, as I said before, these galls, they has practically the same structure and the same tissues. So the male and females, they feed on the same feeding site, because they induct a typical nutritive tissue and the middle portion of the gall.
All right. Moving on to the next question, Bruno Garcia Ferreira. Graciele, very clear presentation. Why do you think vascularization is poorly studied in galls? Are they complicated to analyze?
Thanks, Bruno. I think that is difficult, especially phloem analysis. Identification of the cells of the phloem. Xylem not. Xylem is more easy, but phloem is difficult. That's it.
All right. The next question is to Pedro, and it's a two-part question. I'll put both of them on the screen, all right? Lúbia Maria Guedes Garcia. Good morning, Pedro. Did you do histochemical tests on the galls? And I have observed that in galls with two types of cortex cells, the more elongated ones accumulate secondary metabolites, most phenolics. Is it the case of your gall study models?
Good morning, Lubia. That's a great question. You just blowed my mind with this question, because I've never thought of this before. I did just the primary metabolite histochemical test, because as I showed to you, this gall has a high range of sclerification in its tissues. And since we have to do hand cut, fresh cuts, to do the histochemical tests, it is kind of hard to cut this gall. And this gall is very small. So in this part that I showed you, that the galls differ in the cell, the cells differ, there are elongated cells and globoid cells. It is kind of soft to cut. So I will absolutely do this test. And I think what it will be. Something curious about these galls is that we don't know that they differ in phytohormones. We were able to do the test of phytohormones, but since the pandemic arrived, we didn't have the chance, and the machine that do this test broke. So this is put in the wardrobe for a minute.
All right. René Carneiro asks, Graciele, elegant demonstration of plant cells process. Bio results, do you think your observations corroborate results on auxin levels like the ones presented by Jack Schultz yesterday?
Thanks, René. Yes, corroborate. We detected auxin in tissues of the young galls, especially in parenchymatic cells that differentiated in xylem.
The next question is not directed to any of, I think the ones that talked about the difference of sex, all right? Congratulations to all. Can the difference on the galls female be attributed to the presence development of the insect larva?
Yes, they can.
In my case, the differences is mostly due to I think that I didn't test this, but the difference can be from the differences in lifetime from these insects. So the females, by staying longer, staying with a longer stimuli in the plant, they consequently induces bigger galls than the males. So I think that this is correct.
We have two more questions. Maria Virginia Urso. Pedro, are the immatures of the different galls of different size? Nice study.
Oh my God, that's an honor. Actually, Maria Virginia Urso is the one of the people that described this species, Lopesia matybis. So when I was opening these galls, I saw differences, but these differences in size of the larvae and the pupae of these insects was very slight, very light. It was, I don't know how to say this, but there were differences, but these differences were not significant. But how do I know that the pupae are male or female? Talking with one of the people that described the Lopesia, we could analyze the final instar of the abdomen of the insect, and we could see the apparatus of sexual. But that's a no, Maria Virginia.
All right, the last question is directed to Graciele by Rosi Isaías. Gracie, do you intend to keep on studying vascularization? What are your next steps?
Oh, yes. My next steps is analyze the vascularization in other taxa of gall inducers.
All right, this was the last question. I would like to congratulate all of you and thank all of you to this presentation. There are very good commentaries on the chat. If you would like to know, we will have now a 10 minutes break. Let me put it on the screen, and we'll be back at 10:30 a.m. to the second part of the symposium, right?
Gabriel,
put on the screen.
I just want to thank the organization of this symposium. It is very organized, and I liked a lot to participate in it. So it is a pleasure and an honor to have my references in Gall studies asking me questions. It is very mind-blowing. So thank you all for doing this symposium.
Thank you, Pedro, for your words. And we'll be back in 10 minutes, all right? Stay with us. See you soon.
Hello. Hi. So, good afternoon. I'd like to welcome everyone to the fourth session of the symposium. My name is Sabrina Vasconcelos Caran, and I'm a biological sciences undergraduate student at the Federal University of Espírito Santo. I'm also a member of the chapter in the seção Botânica, and I and Gabriel are going to be the mediators of this session about multidisciplinary approach in the study of galls.
All right. Hello again. Let me introduce the three speakers of this morning. The first one will be Jocelyn Silva de Almeida Cortes. She has a degree in biological sciences, bachelor and licentiate from the Federal University of Pernambuco, obtained a master's degree in botany from the Federal University of Rio Grande do Sul, and a doctorate in biology, plant ecology, Université de Sherbrooke, Canada. She held a postdoc linked to the Federal University of Minas Gerais under the supervision of Dr. Geraldo Wilson Fernandes and senior internship in 2015 with a Dr. Bill Shipley, Université de Sherbrooke. She is currently a full professor at the Department of Botany at the Federal University of Pernambuco. She is currently the coordinator of the Environmental Sciences area at CAPES, where she served as deputy coordinator of academic programs in the area of environmental sciences, associate professor at the Université de Sherbrooke, acting as a collaborating professor and supervisor of the Master in International Ecology and coordinator in Brazil and of the NGO Ecology Sans Fronteiras. She has experience in the area of ecology with an emphasis on ecology of population and communities, acting mainly on the following themes: multitrophic interactions, plant gall-induced insects, plant miners, herbivory, invasive plants, fragments of Atlantic and Caatinga forests. Let's display the good morning, Jocelyn. Now we can see your presentation and you can begin. All right.
Yes. Good morning, Gabriel. Thank you very much for the presentation. I would like to thank also all the organization, Rosie, Dennis, Bruno. For me, it's a pleasure to meet all my friends that loves Gall. And I hope that my talk will, how can I say, a little bit disparate something new or some question for the young students that are just starting or would like to continue working in galls. Okay, I guess we can start with the presentation. We have here just a few minutes. Okay. Divest of galls, dry forest vs rainfall.
Sorry, Jocelyn, we can see only a white screen. It's not showing your presentation.
Well, are you sure? Because for me, it's the same
Yeah, just a white screen.
Continue right screen.
Yes. I'll put Sabrina just if she can say something to help you.
Well, because
Can you send to us?
Yes,
I will put the email on the private chat.
Okay.
Just
Do you mind, Jocelyn, if we put Bruno first?
No,
Before you...
go ahead, please.
All right, thank you.
I will stop the
So,
sharing, okay, and I
yeah,
will send you by email.
all right, thank you. Now I will introduce, sorry, Bruno Garcia Ferreira is an adjunct professor in the Department of Botany at the Institute of Biology of the Federal University of Rio de Janeiro since 2018, 2018. Sorry, he holds a degree in biological sciences, a master's in plant biology, and a PhD in plant biology, all from the Federal University of Minas Gerais, having completed a sandwich internship at the University of León, Spain. He has a degree in botany, mainly in plant anatomy, cytology, and plant physiology, having worked in the studies of morphogenesis of vegetative reproductive organs and galls inducted by animals in plants, the cytological role of oxidative stress in plants, and new methodologies in histology and plant histochemistry.
Good morning. Thank you
Good morning,
for the
Bruno.
presentation. It's an honor to be here with Jocelyn and Dennis. Nice to see both. And now I will share my my lecture. Can you see?
Yeah.
Okay. I am Bruno Garcia Ferreira, professor of the Federal University of Rio de Janeiro, and today I will talk a little bit about our story with Dictylenchus. Our story is with the studies of the development of these galls induced by Dictylenchus galliformis, a nematode, in plants of the family Melastomataceae. This work was conducted by the Gall group, the structure, physiology, and chemistry of neotropical galls, with the coordination of Professor José Isaías from Federal University of Minas Gerais. Then we are able to study galls according their development. We study the ontogenesis, the formation of the plant tissues, and the redifferentiation into gall tissues, the histo and phytochemistry to understand the relationships among the substances accumulated in galls and their functions, and then we may also infer the functional anatomy, the function of these tissues for the gall inducers. We are able to study insect-induced galls, and we compare distinct feeding habits of galling organisms and their impact in the plant development. The study of plant development is important for us plant anatomists because the galls represent the reacquisition of plant potentialities that are not expressed in the ungalled organs, and then the galling organism may induce the new formation, the redifferentiation of tissues. Here we have the final phase of the development of this morphotype, the horn-shaped gall. We have the reacquisition of meristematic features in this region of the gall, where the horns of these galls are formed. And then this gall morphotype may represent a reacquisition of the plant developmental potentialities, which are inside the plant DNA, but are not expressed in the normal organ development. Then, how did we begin to study these galls induced by Dictylenchus galliformis? We became very curious because these nematode galls are induced in aerial parts such as inflorescences and leaves of Myconia species and several species. And nematode galls are usually induced in root plant parts. And these plants are native to Brazil. And this is a peculiarity when we compare to several insect galls, because a single species of galling organism is able to induce galls in several hosts. And our questions were if these galls differ among the host plants, the host plant potentialities influence the formation of tissues of in galls, and how these weird galls are formed, because they have a very strange, very weird morphology. And then we studied the inflorescence galls, and we see that these galls have an indeterminate growth, and this indeterminate growth is not described for any gall types. We saw that most of the meristematic and anatomic features of these galls are similar among the host plants, and they are conservative, maybe because the host plant potentialities are not so quite distinct from another of the same genus. And there were distinctions in indumental type, for example, which are distinct uh, among each host plant species. So the potentials of the host plants are is expressed in these galls. We also saw that these galls may act also in ungalled parts of the inflorescences and other inflorescences in the galled individual, affecting the carpel numbers of the fruit and flowers. And this level of change is distinct depending on the host plant species. Here we have the galls induced in the leaves, and we also see the indeterminate growth activity. These galls may be divided in outer regions, which are not in direct contact with the nematodes, and the inner regions. The nematodes arrive to the plant, the leaf, for example, the meristematic leaf, and then begin to feed in the surface of the leaves, inducing the formation of discovering structures. And the inner region, in direct contact with the nematodes, has meristematic features and is also a nutritive tissue. Then the nematodes feed here and induce the formation of new covering structures, here a young and meristematic emergency, which will form another emergencies, and then this gall continue to grow in all the developmental time, and then several generations may occur inside these galls, and they have a longer life cycle, these galls. Here we have the same indeterminate activity here in the abaxial region, in the lower region of the leaves. Here examples of new galls and older galls. Then in mature leaves, we may see these galls, which are already mature, and then the new formation of emergencies here brings to the formation of this morphotype after some months. So the common storage parenchyma and the typical nutritive tissue of this gall is similar uh, with those of insect galls, but the indeterminate growth activity is a peculiar and was never observed to other galls, including other nematode galls. Then we compared and saw that common storage parenchyma is similar to the other galls, including insect galls, and the nutritive tissues also have features of typical nutritive tissues. We then make other questions on the how these organisms may induce this neoformation, this reacquisition of primary thematic activity in the typical nutritive tissue. Then we had works with ultrastructure, anatomy, immunocytochemistry, histochemistry, and physiology on these goals to respond some things. Then the common storage parenchyma, the outer region, is the region where there is accumulation of starch grains, phenolic accumulation in these tissues, here in the vacuoles of these hypertrophied cells, and we detected cell wall components for stability in this region. And these components and the pectins and hemicelluloses vary according to the host plant species. They have also chloroplasts, which are not so well developed, but they may function and accumulate starch. The outer compartment has accumulation of phenolics, and the inner compartment also presents accumulation of phenolics mainly in cell walls, here also in cell walls, and in nuclei of the typical nutritive tissues. We detected distinctions between this gall in Myconia albicans, here cross-sectioned, and Myconia baguensis. In Myconia albicans, we observed the increased phenolics associated to cell walls, and this result was related to a reduced peroxidation of the lipids. Then these galls are under less oxidative stress and less production of reactive oxygen species. We related this, as other works, to the phenolics, which are antioxidants. In the gall induced on Myconia baguensis, there are increased carotenoids. And then we observed that these galls may have increased dissipation of heat due to this feature. Then the same organism may induce distinct antioxidant defenses depending on the host plant species. They need to adapt to the host plant potentialities, and they use these features to protect the gall tissues and promote a longer life cycle for these galls. The outer gall compartment has also, we detected here in this work, the accumulation of auxins, which is responsible for the cell growth. And then hypertrophied regions, the outer regions, has accumulation of auxins. The inner gall compartment has accumulation of here a strong detection of reducing sugars such as glucose and fructose in this region, and where the nematodes are feeding and new cells are being produced. So these reducing sugars are essential. These galls are very strong sinks of reducing sugars. Here, accumulations of proteins, I think they are enzymes because these cells are in active metabolism, producing new cell walls. Here we have several organelles in these active cells, which are dividing. Then these cells are nutritive and also metabolic active. We detected cytokinins in these cells of the inner gall compartment, which has primary thematic features, producing new emergencies here inside the galls. The accumulation of cytokinins is indicative of hyperplagic layers, hyperplagic activity, and also the more strong sink occurred here by the accumulation of cytokinins, because cytokinins is important for the accumulation of invertases, which are able to break the sucroses into glucose and fructoses, then making available the reducing sugars here in this region where the nematodes are feeding, and also plant cell walls are being producing, cells are dividing, so energy and carbohydrates are necessary for this construction of new cells with new cell walls. Here we have a synthesis. The auxins accumulate in the outer layers, which have hypertrophied cells. The starch is broken by the detected activity of phosphorylase, then starch is broken. Here is the detection of phosphorylase. And in this transition region, they break starch into sucrose, and invertases break sucrose into reducing sugars, glucose and fructose, here available to the nematodes feeding. And cytokinins are important for the indeterminate growth activity, with other signals which we do not know yet, these molecules, but we need to know to understand how these Golgi organisms are able to induce these changes here, promoting the indeterminate growth activity, the maintenance of permeristematic activity. This this Gall system is also being studied by Igor Abarriola in Federal University of Minas Gerais. Here we have a work studying the accumulation of iron and also aluminum in these galls. And Igor and colleagues detected that these galls may accumulate and compartmentalize these metals in distinct cell compartment depending on the host plant species. So the Gall formation here is also dependent on the host plants' potentialities and their environmental adaptations. So this Gall system is very interesting, very excellent model. And I talked to you a little bit on the study of the development of these Galls, and we need to integrate with other scientists to understand step by step the Gall induction and functioning. These nematodes are true and biotechnologists. They may change the gene expression of these cells and maintain the promeristematic tissues. We are step by step studying these goals, and we depend on the collaboration of other scientists, such as you, for example, to understand these distinct chemical and gene expression activities which occur in these goals. So let's work together. I invite you to work together. I would like to thank the Gall Group, the Federal University of Minas Gerais, the financial agencies, and my institution, Federal University of Rio de Janeiro. Here are my contacts. Thank you very much. Thank you, Gabriel.
Thank you. Now we'll show the Jocelyn's video, and then we'll see the Dennis, and then we'll have the round of questions. All right.
Okay.
Let me put Jocelyn on the screen again.
Thank you very much, Gabriel. And before I start, I would like to congratulate Bruno for the wonderful presentation. Was very nice pictures, very nice work. I guess I learned a lot. And just to register, we found also nematode galls in Miconia prazina here in the Atlantic forest, Bruno. So I'm not sure it's the behavior is the same, but probably it would be very interesting to compare. So let's go work together.
Let's work together. I want to see these goals. Igor wants to see these goals.
It's very interesting to accumulate the iron and aluminum. It's integrating. Okay, so let's start the device of galls, dry forest and rainforest. As I say before, I propose to, first of all, I think to propose comparing, the studies between that were done between the dry forest and the rainforest. The next slide, please. But when I was thinking about this talk, I found the scientific scientometry review And it was very interesting because I need to just think in my way, because it's very smaller for me. We have a lot of work in the last years doing everywhere on the world. And in Brazil is not different, but we need to work more. There is, as Bruno just said, so many different points that we need to start, we need to discover, we need to learn with the goals. And if we think about the different environment and how the goals adaptate first to find the host plant, the time, because if you compare the rainforest and the dry forest, we know that the rainforest, the plants will have leaves all the time, but in the dry forest, they lose the leaves. So how the inducers can find what's the time, what is the good time to induce the galls. Então, so Walter Araújo, he did a very nice work when they proposed to investigate what we are doing in the last 30 years of research insects in Brazil. So Walter, he found that the number of the paper increased a lot. And when, the next slide, please. And when they decide to investigate this, he found 1,037 papers everywhere working between 1988 and 2017, especially 182 were done in Brazil. So, of course, the most common topic was in ecology, but also inventory. And if we can think that just few papers, especially in Brazil, that's a large country with mega diversity, it's not a lot. In fact, it's a few papers, and probably because just a small group, now we grow, we are more research interested in the study of the galls, but the dimension of the number of research working in Brazil, south to north, is still smaller, and also the papers are also few. And when we thinking about the different region, we can see that the number of the papers and different paper, doesn't matter if it's ecology, inventory, taxonomy, agriculture, anatomy, as Bruno just show you a nice presentation. We note that we are concentrated in the southeast of Brazil. And this is a good reason. We have the best and the older, I mean, the seniors, the people that start working in galls are researchers that come from the university from the southeast. University of Minas Gerais, Rio de Janeiro, Gianni Kraus that started the working. And this concentrate the number of the papers with the first author or with the people, the group of research from the southeast Brazil. And after that, Central West and Northeast, we are just starting. Thanks that now we see that we have a little bit more research interest in the study goals. North and South, few research. We have Dr. Geraldo that is working a nice job in the University of Rio Grande do Sul. And if we focus, we just have eyes to the southeast, we see that Minas Gerais is a state that we have more papers published. And it's good because Minas Gerais, as we see in the northeast and especially in Pernambuco, that's the states that I'm located, has different kind of environment. So they have also different opportunity to investigate different flora, different kinds of plants. Next, please. Next slide, please. And Minas Gerais, in the north of Minas Gerais, they have a different kind of forest that is a mata seca. It's part of the tropical dry forest. And first of all, we are separated tropical and dry and rainforest. When you're talking about tropical, in general, we're just thinking about rainforest. But we sometimes forget that we have large seasonal dry tropical forest. And this is, we are represented here as the largest one, that is Caatinga. So you see that tropical dry forests are widely distributed, Mesoamerica and South America, but also Africa, Asia, and Oceania. Next, please. When we're thinking about tropical dry forests, especially South America, specifically Brasilia, and you see that even if you have as a distribution of tropical dry forests in Mesoamerica and South America, few papers are produced, are published with data from the tropical dry forest in South America. Marcel Coelho and team, they wrote a chapter of the book called Galls from Brazilian Tropical Dry Forest, Start of Knowledge and Perspective. So in 2014, the author mentions that only 4 studies were conducted on tropical dry forests from South America. And they describe the 4 papers that were produced. And in this chapter, they described that the different papers, the 4 different papers, work and found 29 Gallen species in 17 host plants. And another one, the second one, just 19 Gallen species on 19 host plants. The third one found 64 Gallen species on 48 host plants. And in the last one, just few Galls. Next, please. If you're thinking about those papers, 4 papers, the first thing that I thought is that true? It's just so few work on dry forests in the Brazilian tropical dry forest, just 4. Well, in 2014, sorry, probably the author just found these 4 papers. But in fact, the people were working, the group were starting forming and start going to the field working and looking for the Galls and be integrated and became working with those. So let's understand a little bit about the tropical dry forest in Brazil. We have the Caatinga, and you can see the Caatinga occupy most of the northeast of Brazil and the north of Minas Gerais. And when you look for the first papers that I show you, the Walter Araújo cytometric paper, we can see that in Northeast, we have research that were done in the states of Ceará, Pernambuco, Alagoas, Sergipe, Bahia. As a first, we can think, if the Caatinga cover most of the Northeast, why so few papers are developed in this region? If you look at, you see that the paper concentrate for the materials harvested at Pernambuco State, and after that, Bahia. And one reason that I can see is the university location and the field work, the field sites. It's so far from the people travel from the university most of the time in the capitals that are near of the Ocean Atlantic to go inner of the state and looking for the galls or looking for the inventory of the plants. But next, please take this picture in your mind. And next, please. We see that one paper that were published at Revista Árvore start working with the galls and Caatinga in August 2005 until April 2006. But unfortunately, they published the paper just in 2012. So this one is the first paper or the first work with a Caatinga inventory of the galls. And Carvalho Fernandes and all, they found, please, next one, when they are working in 3 different study areas, evaluate the impact of the anthropic action. The one was anthropic impact, and the other one considered preserving, the third one, intermediate situation. Next, please. Carvalho Fernandes found 25 galls morphotype in 18 plant species with on 10 botany family. And in this case, the work points that the Salpinias was the families that present the number, the 6, especially Salpinia pyramidalis. And the richness of galls was influenced in this case with the degree of the conservation of the study areas. So you see that this paper were not, probably because of the reason of Walter Araújo appointing his paper. In general, most of the galls papers are published or were published in local journal. And sometimes those papers are just forget. The people usually, when they are doing the research, they usually use the information that was published in the international journal. But it's very important, especially in inventory research, that we looking for the papers published in the local or Brazilian journal papers, or even sometimes the paper, unfortunately, is not come to be published, but still as a thesis or master thesis keep on the libraries, unfortunately. So the next one, we also found a second paper published from 3 species of Euphorbiaceae, also describing the galls. Next one, please. And in 2011, Santos published one paper that in this case he traveled field trip when he do in Pernambuco state, harvest different sites, different Caatinga sites, and he found 64 different types of gall from 48 species of host plants, 17 families. Again, in FAPERJ, you say families that presents a number of galls, 23%, falling for Euphorbia, Boraginaceae, Melpiccaceae, and Myrtaceae. So note that is most of the time, the galls that the inventory are detecting is from Cecidomedia. But also, as Bruno just talking about, we can find galls induced by coleoptera, heteroptera, and nematoid, as he talking about. Next one, please. Next slide. So in this, probably more, how can I say, he works that cover most of different types of Caatinga sites, and he found the 6-8 different galls from different aspect of morphological aspect, from different leaves and stems. And this is something very curious. In dry forest, the leaves has a lifespan very short, because it depends of the precipitations, it depends of the rain season. So the animals, the inducer, need to have another option. And in general, we can find a lot of galls and the stems, and why not the roots? Probably we're not looking at this good organ to find find the galls from the dry forest. And after that, we can start thinking about others methodology to start the galls from the dry forest, because you are doing the same that you are doing from the rainforest. And we need to think that it's important to do this just in the rain season, but also in the dry season, and looking for the different organs of the plants, and also the difference even in the as I say, in the roots. Next, please. So we can find here nice pictures with different galls that are published in the Santos et al. Next, please. And after you pass those, we see a new one from Sidemaya describing two new species of Lopecia, a diptera that was associated with Mimosa hostilis. It's a Mimosa Fabaceae leguminosa in Brazil, from a dry region at Caatinga. So you see that we have interesting, but we need people, we need research that would like to work in dry forest. It's not easy. Minas Gerais, as I say, the people from Montes, Unimontes could look for the galls from the dry forests that they have, and also the other resources that are located in the northeast. And thinking about this, next one, please, we can point, and for me, it's the most complete work that I have been notes presented. I have as the opportunity to be part of the committee of Elaine Contrin Costa. She did a master thesis that is very, very nice. And why this masterpiece is so, have so many data, are so complete? One reason, because a Universidade do Estado da Bahia, UNEB, the campus where she studied was located close to the Caatinga. So this is more incentivate for the student to go there and work with the plants and looking for the galls. So if you see the map, the regions that Costa studied was located in the central south of Bahia. And in this region, she had the opportunity to study with 3 different ecosystems, Cerrado, Mata de Galeria, and Caatinga. And she found 500, almost 600 morphotype of galls. It's something that nobody did before. Just in Caatinga, she found 91 morphotypes of galls. So it means that if you work, if you're looking for, you find. So probably all the eyes are just looking for the galls in the rainforest. Of course, the rainforest is close to the universe. So it would be easy for the research go to the rainforest, go to the Atlantic forest, go to the Amazonia. In fact, Amazon is less explored, less studied than the Atlantic forest, then go to the Caatinga. So when Elaine Costa decide to study the galls, decide to do the inventory, she found a large number of morphotype distributed in different families. So at Caatinga, she found 4 order, Fabalis, Malpighialis, and Myrtalis, 3, as the most important. And again, Leguminosa Fabaceae was the most representative of the family, host family of galls, followed by Myrtaceae, Malpighiaceae, Bignoniaceae. If you see Copaifera, or the genus or the species that has most different morphotype, 28. So I am not describing here all the genus, all the species, but one thing that you need to keep in mind, if you're looking for, you find. So let's go to study, let's go to have more time to investigate the, not just inventory, but also the physiology. Can you imagine the adaptation of the plants to survive in short time with water availability? And can you imagine the endurance to find the plants that has adaptation for surviving a short time with water distribution? So we need to think more about to study galls and all the process of the interaction between plants and inducer gall in plants with in dry forests. Next one, please. So as you see, Elaine she finished her master thesis just in 5 years ago. So exactly yesterday, I guess, as 25, that she presented her master thesis, and probably she's publishing the data. But one thing that Elaine, how can I say, she invited Juliana Santos Silva to start a research group. And after that, other students starting or continue studying galls in the same region. So one paper that was published in 18 was Gilda Brito working with the richesse of galls and the Caatinga with different anthropization level. We can see the next one, please. And if you see, she found 20 galls morphotype in 9 plant species, belong to 8 families. So another family can starting showing up, or we are discovering new families that support galls as Annonacea, Bignoniacea, Caparacea, Convolvulaceae, Euphorbiaceae, and Leguminosae in general, we find a lot. So it's mean that we are find more galls in Leguminose and Euphorbiaceae because they are the families that are rich in dry forests, or it's because you are not looking so many times for the others sites and field sites that has different flora, so we can explore more, we can find new, and probably once Dr. Sid Meyer talking to me say, probably we have so many new different insect inducing galls at Caatinga, and we don't know because we need the ecologists, we need the research go there, harvest, study, and of course we need to identify the new species or even new genus, as you can see in the last paper I showed from Sid Meier. So let's go to work, let's go to investigate, because the rain forest, we know that we need to work more, but we have 10,000 more publication from the galls from rainforest. But, and what about the dry forest? What about the adaptation? What about the different environment condition? Is that the anatomy, physiology, biochemistry are close to that ones that we found, we describe from the galls induced in rainforest? We don't know. We need to investigate. Next one, please. So what's the next? In fact, the title was Dry Forest vs. Rainforest. But I was so patient when I discovered that we have so few papers driving with the question, the galls and the dry forest, that I prefer to instigate.
I'm so sorry to interrupt you, but we have other talks.
Yes, so, I close it, Gabriel. I was just saying, so let's go to start as a dry forest. Let's go to working about the subjects. Thank you very much for your attention.
Thank you. I'm so sorry for interrupting you, but
no, but I feel.
Now we will have Dr. Dennis with us. Let me introduce him. Dr. Dennis Coelho de Oliveira is an adjunct professor at the Federal University of Uberlândia, Institute of Biology, PhD in plant biology by the Federal University of Minas Gerais. I had to mute you because, sorry. He has experience in the field of botany with an emphasis on anatomy and plant development, working mainly on the following topics: plant anatomy, plant morphology, cytology, plant physiology, botany, insect galls, histochemistry, immunohistochemistry, accredited in the graduate program in plant biology and in the graduate program in ecology, both from the Federal University of Uberlândia. So, Dr. Dennis, display your presentation and you can begin.
Thank you for introducing me. Firstly, my pleasure share this section with Jocelyn and Bruno. And of course, and first I would like to thank the Anatomical group for organization of this symposium. Especially I would like to thank you, Bruno and Rosie, who work hard to make this symposium realize. Thank you. So I was wondering how I could contribute with this symposium, especially after excellent presentations until now. So I thought, maybe I can talk to you some students showing some aspects of the fascinating Gall world. I have tried to focus my presentation on how dazzling is the interaction between galling organisms and the host plants leading to gall formation. Yes, there's no doubt. Galls are one of the most fascinating biological systems in nature. We can say that because different researchers from different areas can use different techniques to answer several questions in galls. This is amazing. Interdisciplinarity, morphology, histology, chemistry, genetics, cellular biology, and ecology, natural history. We have a huge range of possibilities. But in a general way, galls are a product of host cells and tissue changed by a phenotype manipulator, the galling insect, the galling organism, generating a high diversity of shapes in a very specific interactions. As an example, the superhost Copaífera Langsdorff with different gall morphotypes induced by different species of cestomide. Copaífera Langsdorff hosts 9 gall morphotypes induced by unidentified species of cestomide on this study. The host species showed a classical phenological behavior species from Cerrado, like a Brazilian savanna. They lose their leaves in the dry season, April to August, and the leaf sprouting starts on the beginning of wet season. So, water availability can define the abundance of gall morphotypes at the beginning of September, for example. Most of these inducers have, for example, a univoting life cycle synchronized with the plant phenological behavior. That is, one cycle along the year. Another similar example are the galls induced by Galococcus isaias on Pseudobombox grandiflora and Bistracoccus mataiba on Mataiba guinensis. Pseudobombox grandiflora lose your leaves during the dry season, and the gall and insects shelter itself into the bark depressions of the stem for overwinter. But they cannot induce galls on the stems. But when the new leaves arrive, the galling insects move to induce leaf galls. However, Bistracoccus mataiba, other ecococcidial species, can induce a rudimentary gall on the stem of Mataiba guinensis before overwinter. A dazzling strategy is not. Both species of galling insects have a univoting life cycle. In a rare example in nature, a single species of galling insects, Pseudophacopteron longicaudatum, an emitterm, can induce similar galls on 2 species of Apocynaceae, genus Aspidosperma, Aspidosperma tomentosum and Aspidosperma macrocarpum. These host species occur in symmetries and show a similar phenological behavior with leaf sprouting in September, October. These galls, these galling insects, both of them show a univoting life cycle. These galls are induced in the beginning of the leaf sprouting, related to with water availability. However, some superhost species, as Mimosa gemulata, can bear different Gall morphotypes induced by different species of cestomide. We can show here 6 Gall morphotypes. Most of them with multivoting life cycle, as reported on this beautiful diagram made by Elaine, showing 6 Gall morphotypes. So, different strategies to explore the host resources depends first on the availability of reactive sites, synchronization of host Gall life cycle, especially in seasonal environments, as occur in Brazilian savanna. After that, we have early events of insect plant recognition associated to salivary compounds of the Gall, followed by the production, for example, of reactive oxygen species in the host tissues, leading to biochemical alterations, plant responses, and formation of Gall phenotype. RUS can act as a signaling molecule for growth and development, as have been described in literature, and biotic stress. Here, in an amazing example, the authors showed a time hierarchy of consecutive events detectable in the plant tissues which are initiated by the feeding activity, a free-living herbivore. The early measurable events on the plant tissues are membrane depolarization, followed by calcium changes in intercellular calcium concentration, and the first molecule formed is hydrogen peroxide, a reactive oxygen species. Hydrogen peroxide, this first molecule in free-living herbivore interactions, before that we have phytohormones detections in the tissues. The interactions between ROS and phytohormones, especially auxins, have been described, detected histochemically in Galls and biotic stress. Here we have reported in Galls being detected histochemically and associated with cell division sites, elongation, and essential steps for Gall development. The cell division, growth, and complexity, Galls with distinct tissues and functions, for example, may depend in addition to how long Gall feeding inside the Galls, and of course, the development of new meristematic sites in Galls, as we reported here in Galls induced in Copaiferal Langsdorff by identified species of cestomide, horn-shaped Galls. We can see meristemoid-like cells here. In fact, Galling organisms, Galling insects can manipulate the host tissues at different levels, as a phenotype manipulator. At cellular level, for example, the phenotype manipulator can change the cell wall of the host tissues, as we are studying in different Gall systems using monoclonal antibodies. This change in functional features of Gall associated to changes in cell wall composition, especially pectins and hemicelluloses. Hemicelluloses, for example, is a sugar that can support Gall in diet, as detected and discussed here for Bragança and co-authors. So, in the root node nematode Galls, we looked for understanding changes in cell wall composition can determine infection and die of the nematode Galls, especially the impact on the giant cells, for example, the sites where the root node nematode feed. Yet, using isochemical tools, we can help to elucidate the role of cell wall components in plant biology as a whole. Here, where different changes in pectins of high and low methylesterification, maintaining the cell growth, cell elongation, especially by the formation of giant cells. In addition to isochemical tests, Hymen Microscope, for example, is another useful tool. And here, in Gall-induced Mataiba guianensis, we detected lignins using this technique, and young Galls, blue points here, blue dots here. Why? Maybe because lignins can work as stress dissipation, Some galls have a shelter of lignified cells in the cortex. These lignified cells can be a response of high oxidative stress induced by galling insects, favoring gall establishment. Among these phenol-like derivatives, lignins are the most affected by oxidative stress. The biosynthesis depends on the generation of ROS. So, lignins as a phenol-like derivative that can be manipulated by the galling insects in gall tissues. Most of secondary metabolites in galls are phenolics, detected histochemically in the outer cortex, as shown here. The function of these metabolites seems to be, The function of these metabolites seems to be associated to protection against oxidative stress, maintaining the redox homeostasis in gall tissues, as discussed by Isaias paper some years ago. Secondary metabolites can be too associated to color variation in galls. Anthocyanins, for example, is a secondary metabolite derivative, as we can see on globoid galls induced on qualia. Here, the light exposure determines the red coloration in globoid galls. Galls induced by cestomide on qualia occur on abaxial, adaxial, and this occurrence are submitted to different light condition exposure. Adaxial galls, above galls, are red, while abaxial ones, below galls, are green. In addition, some angled leaves is possible to observe gall displaying two colors. Look in this graph, above and below galls, different colors and different exposition to light conditions, and the angulation can determine the display of galls. Here, to measure anthocyanins, we use spectral reflectance partners to access this pigment content by calculating anthocyanin reflectance index. In our study, we also showed another galls on qualia parviflora, the infralaminar galls. These galls showing no color variation between above and below surface. This demonstrates that in the same host plant, anthocyanins accumulation via line exposure may also not occur, suggesting each gall related to its specific galling has its all anthocyanin metabolism. Another amazing example are Rojas urbelandensis induced galls on the ovules of floral buds on Camissois. These ovule galls developed and works as a physiological seed, stimulating the growth of the carpels, leading to the formation of fruit-like gall structure. But these fruit-like galls may show different colors, dark or red. Why? We don't know what explains these different colors yet. But we can suppose different light exposition like in galls on qualia parviflora. Another example, gall induced by paleomysteroid gall, is an amazing example on Macaria radula. These galls are big, with around 3 centimeters of diameter. These galls have a true nutritive tissues accumulating lipids and proteins. In addition to that, the globoid galls on Macaria radula are attacked by different natural enemies, and these attacks does not depend on coloration. As you can see, different attacks and different display of colors in these galls. We have a huge color variation between green and red. Why this occur? Why globoid galls on Macaria radula show a huge range of colors? Maybe the concentration of anthocyanins in the site of apposition matter to explain this color. May the salivar compounds in the galling insects induce cytokinins, produce cytokinins, and producing sugar, and the residual of a lot of zinc of photostimulated can't accumulate anthocyanins? We don't know yet. There are just some hypotheses we are testing. So, We can see here a very red coloration in these galls induced by Macari Radula, with a lot of trichomes, projections. Is really a very beautiful system. But now we are testing some hypotheses to explain why these galls is red. We don't know yet, but as soon as possible we can answer that. For a while, unpublished that yet. So, in a simple conclusion, galls are one of the most fascinating biological systems in nature. There is no doubt about that. Thank you all. Thank you the agencies for financial support, despite of deep crisis in science in Brazil. But I would like to leave a message for us. We will resist. There is no doubt about this. And of course, thank you my wife to take care of kids while I can participate of this symposium. I promise, Anna, last week I will cook every day. Thank you.
Thank you all. Thank you, Dennis, Bruno, and Jocelyn for the amazing presentations. Now let's proceed to the questions. The first question is from Jack to Bruno. And I forgot, as your time is short, it would be great if you are succinct in the answer the questions, please. Thank you. Bruno, fascinating presentation. As I said yesterday, the cell tissue specific studies are crucial. And so is interdisciplinary collaboration. And then he asks, Bruno, have you thought about alternative functions of antioxidant compounds? The phenolics, for example, could be antipathogen or components of newly formation cell walls.
Yeah, Jack, we need to, this is the objectives of my presentation, to propose more integration among the scientists, understand the development on the anatomical point of view, and later, for us, later with your collaboration, for example, the study on the molecular changes in goals. So about phenolics, yes, we consider it because in our work in plant biology, we correlate the same places where the phenolics accumulate are the same places where auxins accumulate, and they may function as inhibitors of IAA oxidases, the enzymes which break the auxins. So this is one of other physiological functions, but they also may be antipathogen compounds, because these galls induced by nematodes, because the nematodes are very, very small, we didn't observe parasitoid, for example, but there are fungi that live inside these galls, especially in the regions where the nutritive tissues are already senescent. So I think the phenolics are very important in these galls, which are with a longer life cycle, to protect the tissues against pathogens, for example. But you need to evaluate this. Thank you for the question.
The next is from Rosie to Jocelyn. Good morning, Jocelyn. I consider that Araújo did not evaluate the amount of papers of insect gall anatomy and development in this work. Could you realize that?
Hello, good morning, Rosie. Yeah, for sure. I did not see the Araújo in his papers. anatomy papers include. I guess we need to do another cytometry studies that we can include all the others paper, because as you see, I guess it's miss some papers, especially that ones that are published in Brazilian journal. So a lot of things that are published were not included, for sure. And anatomy, I guess, is one of the areas that are publishing, as Bruno and Dennis show. We have a lot thing to do, and it's so amazing, the interaction between the gall-induced and the plant tissues. And you are doing a school, very nice, good research group.
Okay, the next is from Rosichu. She asked Dennis, why do you consider there are so many variations in the color of galls?
It's a really good question. Maybe we have a lot of galls, the same gall morphotypes displaying different colors. First question we do here: what the ecological what the ecological perspective from this red or green or different displaying colors? But before that, we need to understand why these galls have different colors. There is several hypotheses discuted on that. The Fabricano hypothesis, when some galling insects can put salivary compounds like cytokinins produced by the galling insects, induce a big sink of photosimulators, and the anthocyanins can be a residual production of this sugar metabolism. We have the light exposure by stimulate anthocyanins biosynthesis. We have a discussion of some aphids, for example, have some genes associated to determinate color, and some salivary compounds can induce these galls. So there are different galls with different colors, and we need to know what the ecological advantage or not about this.
Okay, the next is from René. He asks Bruno, great informative talk. Do you think there are different nutritive cells profiles and dynamics? Why the nematode nutritive tissue layers are so narrower than those of most Cecidology?
Thanks, René, for your question. Yes, they generally, the nutritive cells are usually more active, metabolic active, so the organelles are more abundant and the nuclei are hypertrophied. I mean, they are larger than the nuclei of the storage cells, for example. But among the nematode galls, the nutritive cells are distinct. Some of them, as Dennis showed us, meloidogen have giant cells. Other may promote cell wall degradation, forming syncytia. And these galls in Dithylembus galliformis have nutritive tissues with the other feature, the peculiar feature to produce not only more nutritive cells, but also vascular tissues, new emergencies, which will cover the other generations of nematodes inside this gall. And why do nematode nutritive tissues layers are so narrow? Well, I think this is because their stylets are shorter than stylets of sucking insects, for example, Cecidology or aphids. But I think in Cecidolmid galls there is variation, but because some galls have very thin layers of nutritive tissues. The galls with several layers of nutritive tissues are usually those galls of Lepidoptera and Hymenoptera, which have larvae with chewy mouth parts, and they are more voracious feeders than the nematodes and the Cecidology. I think that's it.
The next is from Valeria. She asks to Jocelyn, do you think that we should use data from dissertations and theses? Many editors do not accept their citation.
Oh, good morning, Valeria. Yes, when I talk about the number of the researchers, I was thinking about that we have so many more things that we did but not published that keep on the library. That is most important. First, the people publish, the students, the research publish the data, but when you are doing inventory of the number of the studies and to do or to know how the situation of the studies in Brazil or by region. So at this point, so that I suggest to also consider the thesis and the dissertation, thesis, master thesis and master PhD, but just to know the situation of the research, not to include as reference to publish in the international paper. And also because one thing that I noticed when I was preparing the presentation, in general, the people prefer to use the reference, international reference, and keep going this all the time, and don't use the local reference. So those could be forget, just in this was a view that I was trying to discuss this subject.
Okay, thanks. From Rene, yes, Dennis. Can the biosynthetic pathways can crosstalk with other ordinary processes involved in Gall morphogenesis and sometimes be simple side effects of other chemical battles?
Yes, this cross-talking can occur with other different processes involving as Gall morphologies. It's in the case to study each Gall system because they can be submitted to different environment and metabolic changes in the Galls at different times. We have tested this cross-talking, for example. We have measured sugar because the fabricational noise hypothesis, but we can find at now no relation into sugar accumulation and red coloration in Galls by anthocyanins, for example. We have Missouri Gall light disposition using a quantometer to measure light incidence, and in some cases we have discussed that we have provide the exposition of light induce more anthocyanins. But I think each Gall system for a while needs to be still separated because we have no partners yet. Why? Because we have red or green Galls in the same case. But yet, this pathways can crosstalk with other physiological metabolism in Galls.
Thanks. From Lubia, she asks Bruno, great job. Many insect and nematode galls have endoplasts with drugs. Any no-hole of drugs is in galls? Or do you think it is a feature of the host organ that is kept in the gall?
Hey, Lubia, nice to see you again. Yes, I don't know the roles of the druses, and a reviewer asked us about this, and I made a review in the literature on this, and some galls have much more juices and other less juices. I don't know, but uh, there there are hypotheses that juices are or other calcium crystals are the sites of accumulation of the excess of calcium, which is an important molecule, as Dennis showed us, signals in distinct metabolism processes. Then I think that they are a storage way, a way to storage or a way to compartmentalize the calcium in the plant cells. And I don't know the functions against, for example, some authors use it to associate the accumulation of druses as an anti-herbivore feature, but it's not really tested in the most of the plants and for the galls too. So I think it's only a physiological manner to store the excess of calcium.
Okay. The next is from Rosie. She asks Jocelyn, topping the hypothesis that galls are more abundant in dry environments, after analyzing these papers in Caatinga, do you consider it is true for your flora, our flora? Sorry.
Jose, I guess one thing that is could be it's we are subestimating the number of galls present in the dry forest because we have less study. So if we have more inventory, we have more environment, different Caatinga, different part of the Caatinga, because they are present in different state. And as I show the map, we have just one paper published that we found from Ceará, and now we are having studies from Bahia and also Pernambuco. So we need to study a little bit more. When we study more, we discover more, we publish more. So I believe that we are subestimating the number of galls at the dry forest, specifically in Caatinga. So I believe that we have much more different galls, species, inducers, and abundance also.
The next is from Rosie too. She asks for days. Do you have any hypothesis of the destiny of the inducers associated to reproductive organs when plants are in vegetative phase? Where should we look for them?
I'm sorry, my son is knocking the door and I'm distracted. Okay, Rosie, maybe the alohojas are an example. Be on a soil, I don't know, because we collected the samples in the swamp vegetation and veredas, and I really don't know. But Pablini will solve these questions as soon as possible. I promise.
Next, from Igor to Dennis too. Nice lecture. Sorry if I lose the information. You detect the compounds which may provide the red coloration in the Macaria hadula emergence.
Yes, Igor, we detect the anthocyanins in the emergence of Macaria hadula using an equipment like a hand spectrometer. We measure all spectra of light, and then we can detect the peak of absorbance of anthocyanins and then calculate the anthocyanins index.
Okay, the last one from Lubia. Dennis, thank you for the presentation. I hypothesize size that in the Chilean Mediterranean climate, it could be due to different levels of exposure to UV radiation. Have you considered this hypothesis?
Thank you for the question, Nubia. Yes, we consider we just to test this with have an excellent biological system, qualia, because we have goals induced and above and below goals, shader or not. So we measure the light exposition using a quantometer. And so in this paper, we consider this exposure by UV radiation and light as induction red coloration. But how I can say that here, we can be carefully with this hypothesis because we can corroborate this hypothesis in the on goals induced on qualia, but in the intralaminar goals induced on qualia, we can different light exposition cannot induce red colorations.
Okay. So thank you all for the great presentations. We have a lot of comments in the chat, so you can take a look. And thank you all.
Thank you. Thank you for the comments.
Now we have a break, a 5 minutes break, and we will come back with the final lecture, which is entitled Another Brick in Gall Walls. So see you soon.
Thank you. Thank you, Sabrina, Darcy, and Dennis. See you soon.
Hello, good morning. So now we're gonna move on to the final lecture, which is entitled Another Breaking Gall Walls. And the speaker is Rosi Mary dos Santos Isaías. Rosi is full professor at the Federal University of Minas Gerais, has a degree in biological sciences from Universidade Santa Úrsula, a master's degree in science, botany from the National Museum, Federal University of Rio de Janeiro, and a doctorate in sciences, botany from the University of São Paulo. She is currently a CNPq 1B Productivity Fellow, a member of the Brazilian Botanical Society of Brazil, Regional Minas Gerais, Bahia, Espírito Santo, SBB, and of the Education Committee of the Regional Biology Council, 4th Region. She coordinates the graduate program in plant biology at Federal University of Minas Gerais, Note 6, CAPS, from 1215 to 1219. She works in the field of botany, which an emphasis on anatomy and plant isochemistry. The following lines of research stand out: plant development, Gall anatomy and histochemistry, and plant cell responses to the action of Galling organisms. She is interested in environmental interpretation strategies and botany teaching. Now, Rosie, the speech is yours.
All right. Good morning, you all. I hope you can enjoy my lecture that is in a video. And after the video, I'll be here so we can talk a little bit more about goals. Okay? See you. What happened? And the Vice President of the second Symposium on the Biology of Gall. So, return the sound a little bit, very much for your kind of attention, collaboration, and participation in our meeting. My talk to close this meeting is entitled Another Brick in Gall Wall. And when I started thinking about this talk, I imagined the encounter between host plants and their gall inducers. And in this situation, the plants must react. What do I do with this alien? I must try to kill it, but I must survive as well. The first line of reaction of plants to the attack of these galling organisms is the use of phenolics, because phenolics have the ability of scavenging reactive oxygen species which accumulate in gall induction and developmental site. As you know, I am a plant anatomist, and I'm also interested in plant histochemistry, in labeling and staining the accumulation of compounds in tissue compartments. Histochemistry is quite interesting, not just because I can see what is accumulated, but where the molecules are accumulated. And you can see that I show you an example of plant tissues submitted to staining with three-line diaminobenzidine on the images put on the right side of your screen, compared to blank sections placed on the left side of your screen. In this image, you can see the higher intensity of staining of the cells in this gall, in chlorophyllous parenchyma, where photosynthesis is happening, in vascular bundles, where the translocation of accumulates is occurring, and also in the cells nearby the nymphal chamber, which are the cells closer to the galling organisms. The reaction of 3,3-line diamidobenzodiene indicates the accumulation of hydrogen peroxide, which is one of the most common reactive oxygen species accumulated in plant tissues. An important point regarding the translocation of phenolics to Gall induction and developmental site to deal with the accumulation of reactive oxygen species is that phenolics will not only interact with ROS, but they may also interact with other molecules. For example, indole acetic acid oxidases. When phenolics block the activity of these enzymes, the level of indole acetic acid, one of the most important phytohormones, will increase. And plant tissues and cell responses to this metabolic variation will be cell hypertrophy. And cell hypertrophy is one of the main processes we detected in Gall developmental sites. Here, I illustrate the histolocalization of phenolics and oxines, phenolics using iron chloride and oxines using Ehrlich's reagent, and you can see that they are detected in similar size in the lenticular gall on Piptadenia gunoacantha. So this overlapping of phenolics and oxines indicated, according to our interpretation, the interaction of these two molecules in the process of cell hypertrophy. We could also imagine that the overlapping of histochemical detection of phenolics and oxines had occurred in the lenticular galls by chance. So we decided to repeat the tests using the other gall morphotypes associated to the same host plant, I mean Piptadenia gunoacantha, Fabaceae. And we found similar results for the overlapping of such compounds, as you can see here. The pink reaction for the Ehrlich's reagent detecting oxines, and the brown-black accumulation of phenolics in similar gall tissues. Okay, well, going on, we decided to validate our histochemical detection of phytohormones using immunocytochemical tools. And we could also put another missing brick in this wall. I mean, we could detect the accumulation of cytokinins both by histochemical techniques and also by immunocytochemistry. So here you can see in blue sites of hyperplasia, I mean intense cell divisions in the lenticular gall morphotype on Piptadenia gunoacantha. And you see that these tissue sites are different from the tissue sites where we could detect auxins. And in green fluorescence, you can see here in the lateral parts of this image, the labeling of the epitopes of transzeatin, one kind of cytokines very common in plant tissues. We also used anti-IAA, a polyclonal antibody, to detect, to validate the use of the Ehrlich's reagent for histochemistry. So in summary, we had sites of cell hypertrophy detected with Ehrlich's reagent and sites of hyperplasia detected with silver nitrate, bromophenol blue, and the relation with the immunocytochemical detection of the epitopes, both of auxins and cytokinins in their specific sites. I assume this is an elegant result for the two process, for the histolocalization of these two processes we are constantly reporting in Galls. I mean, cell hypertrophy and hyperplasia. And the mosaic of these sites is determinant for the development of this variety of Gall shapes we find in nature. Some other important targets of our investigations on the cytological responses to the attack of the galling herbivore are the cell wall components, and we assess these components by staining with chalcofluor white and monoclonal antibodies. And you can see here that we had the results for cellulose microfibrils, hemicelluloses, pectins, and two types of proteins. And I will illustrate this for you using Inga ingoides, a Fabaceae species, which is associated to 3 galling herbivores, and this association results in globoid, lenticular, and fusiform galls. And I will illustrate here the labeling of cellulose and hemicelluloses in the fusiform galls. Okay, cellulose microfibrils are responsible for the remodeling of cell walls, so the axis of cell elongation and the transformations in shape demands this remodeling, and we could detect it using the chalcofluor white. Regarding hemicelluloses, we expected this situation, this scenario. We expected the hemicelluloses to be less detected in young Gall stages of development, and as long as the galls go into maturation, we should expect stronger labeling of the hemicelluloses, just because the primary role of these hemicelluloses is to anchor the cellulose microfibrils. But what we detected was not exactly these. So we will see that there are some other possible functions for the hemicelluloses, as for instance, additional carbohydrate reserves. So here you can see in blue fluorescence the labeling of cellulose microfibrils by the chalcofluor white, and in green fluorescence the labeling of xylans with the monoclonal antibody LM10, which is specific for the epitopes of xylans, and they were detected so in xylem cell walls and in fiber cell walls. I mean, xylans are directly attached to secondary cell walls, and they help in the support, in the mechanical support of the galls, while LM15 is the antibody specific to the epitopes of xyloglucans, and they were detected in phloem, for example, in the parenchyma, and also in the nutritive tissue. And that's why we interpreted the xyloglucans labeling here as an additional carbohydrate reserve to the galling herbivore. In the case of Inga ingoides, we had also expected to detect heteromanans, but the results were negative. In another Fabaceae species, Mimosa tenuiflora, we could get to positive labeling of epitopes for heteromanans. And why am I talking about heteromanans? Because heteromanans are hemicelluloses common in seeds, in tissues of seeds of Fabaceae species. So I expected them to be also labeled in the nutritive cells. But in Ningengoids, it does not occur. In Mimosa tenuiflora, it occurred. So let's wait for the scenes of the next chapter. Regarding pectins and proteins, we have more results, and we could see that pectins labeling may be related to the rigidity of cell walls in outer tissue compartments, in tissue cells in the periphery of the galls, and pectins may be linked to the porosity of cell walls in inner tissue compartments. I mean, in cells next to the larval chamber. This porosity is important for the translocation cell to cell, translocation of molecules cell to cell, while the rigidity is important for the mechanical support of the gall. Regarding proteins, they are also important in the case of extensins to the anchorage of cellulose microfibrils, and that seems to be what we can interpret in the cells of galls, while the AGPs, the arabinogalactan proteins, are usually related to the avoidance of programmed cell death and to the avoidance of hypersensitive reactions. But the labeling of AGPs are not so constant, but they remember they could be an important tool for plant cells to become alive. And here I will illustrate, just really to illustrate the results with pectins in galls induced by Ditilencos galliformans, a nematode on Miconia albicans. And here you can see the fluorescence of high methylsterified homogalacturonans in non-gold leaf tissues, very intense, and a little bit less intense in the tissues of the nematode gall. And to finish, I'd like to tell you some brief results on the symplast, where we can see the differentiation of plastoglobules, multivesicular bodies, lamellar bodies, and lomosomes. Plastoglobules are very important because they also are involved in the scavenging of reactive oxygen species, and they are these corpuscles differentiated inside chloroplasts. The multivesicular bodies, prolamellar bodies, and lomosomes are involved in the rebuilding of endomembrane systems. So plastoglobules control the oxidative stress, while these other structures rebuild the endomembrane system. So they are quite important for maintaining the health of plant cells under the attack of the galling herbivores. I would like to thank the governmental agencies for financial support of our studies of research lines, FAPERJ especially for the financial support of this meeting in the remote mode. I hope we can be together and count on FAPERJ for our next versions of this event. Iniciação Botânica, the group that is kindly helping us in the mediation of this event. The second Symposium on the Biology of Galls organizing committee for being so kind, so efficient, so professional, and always doing this and that in a very kind way. It's the word. I'd like to thank my collaborators and the students that are together with me forming the Gall group in the Federal University of Minas Gerais and the other institutions trying to put all the pieces of the great puzzle together. And I'd like to say that all in all, it was just bricks in the wall. Thank you very much for your kind of attention. Hope to see you in the third symposium on the biology of galls. I hope in presential mode. Okay, see you.
All right, all right, all right. That was a great speech. Thank you, Jose. Congratulations. That was a pleasure to hear. And we have a lot of questions on the chat. Anna would like to introduce yourself. The first time you're here today.
Yes, I am Anna. Good afternoon, everyone. I'm also
We
a
can hear
biology.
you. We can hear you. Sorry.
I'm muted?
Try it again, please.
I'm not muted. I don't know.
All right.
Listen to Anna. It's okay.
Can you hear me?
The first question is...
Gabriel, we can listen to Anna. She's
Sorry?
okay. Anna is okay. She's on. She may present herself.
Okay, I'll start again.
Okay.
Good afternoon, everyone. I'm Anna. I'm a biological sciences undergraduate student at the Federal University of Minas Gerais in Belo Horizonte, Brazil.
Right, the first question is on the screen. It's by René Carneiro. Beautiful complementary histol localization of auxin in cytokinin. Jose, have you ever thought of trying to localize the hormone transporters? They must be distributed in gradations to generate this.
Well, René, no, I have never thought about that. And you know, it's not that easy to work with this histolocalization of fit hormones. We were happy to find some good models where we can have positive histolocalizations. As you saw in Bruno's presentation and here with Piptadenia's calls, but we can try. First of all, we must test the protocol, and probably we will find a good model. As you can see, it's easier to detect these fit hormones in galls than in shoot apex or root apex, for example. Sibeli Bedet tried a lot to establish the protocols, and the staining were quite diffuse, and in galls they are much more intense. So it can be a step to follow. Okay.
It's muted, your microphone.
Next question, Jose, from Dennis. How do we deal with cytokinins on the histochemical tests? These class of phytohormones can be quickly degraded.
Oh, yes, Dennis. Most of the times we use really fresh samples, okay? And in the case of auxins, we must prepare the sections and immerse in a fixative, EDAC, to fix the auxins. But for CKs, for cytokinins, this prefixative cannot work out. So the most good results we have are with very, very fresh samples, okay, and handmade sections. If we try to fix and include in paraplast, PEG, polyethylene glycol, or historesin, we cannot have good results. all right? Just because the fetal hormones are degraded.
Lúbia Maria Guedes Garcia asks, Professora Rosi, the production and accumulation of ROS in photosynthetic galls should be greater in the inner tissue under the influence of the insect or in the outer tissues due to photosynthesis.
Well, Lubia, nice to see you here. I miss Chile and our encounters a lot. Well, as you could see in the example I illustrated, it's quite common to detect this overaccumulation in tissues with high metabolic activity. So in general, we can detect in photosynthetic tissues, except in galls that do not have photosynthetic tissues. In tissues near the gall chamber, nympho or larval chamber, and also near vascular bundles. These are the three most common sites of accumulation of reactive oxygen species. Okay, so it depends. If the gall has well-developed photosynthetic tissue layers, we will detect the reactive oxygen species there. But if it's a light green, yellow gall, no, the overproduction of reactive oxygen species will occur next to the chamber, right?
Okay, next question from René. To establish well-delimited boundaries of histolocalization like that, there must be repressors and degraders of hormones at specific sites. Great model for future molecular investigations, isn't it?
Yes, yes. Very good, very good. Up to now, I don't have an idea of how can we go on, but we can try to establish a map of possibilities, okay, to go on and try to find out why, at least some tips on why are these phytohormones accumulating in galls. And you mean we must use galls under growth and developmental phase, okay? Because if we get a gall that's mature going to senescence, of course, we will not detect these phytohormones. So, good idea. Take it in mind. Let's try to establish the protocol.
All right, this was the last question on our chat, but there are a lot of compliments to your lecture. I'll put some of them on the screen just to see, just to you see the faces of the people who
Juliana,
were.
nice to see you here. Juliana is one of the Powerpuff Girls of the Caatinga. We count on you to improve the knowledge. Thanks, Dennis. I think these compliments are for all our events. I am quite happy with your participation, with the questions and the discussions we had these 3 days. Unfortunately, we are getting to the end.
Now I'd like to invite Bruno Ferreira, the president of our symposium, to say some words to finish our event.
So first, Rosie wants to make a presentation now.
Is it with me or with the girl? The guys?
I can put it.
I can put.
Okay, now
may you introduce?
just stop it so I can say a few words. This video is. Have you ever heard about Aleppo calls? And it's a gift. Professor Jane Krause, who was my advisor, bring us. So just before finishing our event, I would like to show you some curiosities about this gall. It was prepared with great care for Jane, I am sure. Please, Gabriel. I don't know who's going.
So,
Thank you very much, Jane, for everything.
So now I will invite, there is someone more of the organizing committee for our final words on this amazing event. I am very proud to be in the organizing committee of this event. And now I will show some information about this event. As you see, we had people from around the world. I'm very happy to see all of you making questions, interesting in Gall biology. And now let's see a little bit. First of all, I would like to thank the Foundation for research support of the state of Rio de Janeiro. And I would like to thank also to Marcelo Cubo for the arts of this event, these amazing arts of this event. Here I have a cloud, a word cloud with the institutions with participated here, the people from distinct places of the world, Canada, uh, Brazil, Rio, Minas Gerais, UFRJ, Viçosa, Mexico, and so on. And very diversified public. Here, the countries, people from Brazil, USA, Chile, Argentina, Canada, Germany, Kenya, and Colombia. And I make this presentation very quickly, so some countries are missing here, but here is represented our diversity of people. And I would like to thank the organizing committee, and with me, Rosie, René, Dennis, Marcelo, and Valeria, for helping us in the organization of this amazing event. The lecturers around the world, Adriano, Barry, Christian, Dalva, Daniel Burkhardt, Denis Oliveira, Gilson, Jack Schultz, Jarcilene, Kevin Float, Milton, René, Rosie, and Valéria Sidmaia. And I would like to thank our institutions too, because they are our support. Besides FAPERJ, with the financial support, we have the Federal University of Rio de Janeiro, Federal University of Goiás, Federal University of Minas Gerais, Federal University of Uberlândia, and the courses which are involved in this activity. We have several students seeing here, watching this event, the graduate program in botany in Federal University of Rio de Janeiro, the Institute of Biology of the same university, the graduation program of plant biology in Federal University of Minas Gerais, in ecology of Federal University of Uberlândia, and zoology of National Museum of the Federal University of Rio de Janeiro. We had 191 subscribers and 17 video posters, amazing video posters. I loved it. And our channel had several visualizations as expected, and then I am very happy with the where our information, our studies, our science is reaching by now. Here, the peaks of visualizations in the 3 days, and here is showing how our event is a success, was and is being now a successful event. So that's it. Thank you all for being here. We have some, have we other people from organizing committee for the final comments? Okay. Or Ana Maria? Rosie, do you want to talk?
Rosie would like to say something about.
Okay. Are the other guys here? Valeria, Dennis, and Marcelo, and René? I don't know if they are. Okay. So I have no words to say how happy I am. I am expecting now that we can be together in the near future. In 2022 in California, we have a Let's Go event there, this fourth annual event on the biology of galls. And I hope the students can come together with us. Uh, Jane Krause has sent me a message here in WhatsApp that she's crying. So it was not our intention, but we know that you should cry because we are crying also. But we are really, really happy. Thank you very much for the guys from the Iniciação Botânica for getting here with us. Okay, it was a challenge, but you did it successfully. I thank Bruno for being our president, very brilliant organization, and the other guys, René, Valério, Marcelo, and Dennis for all the support. For our audience, thank you very much. I hope we can put some bricks in the goal walls on your mind. So come with us and keep on studying goals. Okay? See you soon.
Just another word, because I was making this presentation by now, and I will place the Instagram of Iniciação Botânica, but I haven't time. But I will invite you to subscribe in the channel, in the Instagram of the Iniciação Botânica, and thank you very much, you, Gabriel, Ana Maria, João, and Sabrina, I'm sorry, and that's it. I think I forgot someone. No, it's okay. Very thanks, guys.
There is one more guy who did not appear here, Wally. Wally Everett is in the backstage, but thank you very much, Wally.
Thank you, you all, Valéria, René.
Can we end it?
Do you want to say something?
I'd like to invite everyone to follow us on social media, Instagram. It's on the screen. It's passed on the screen how you can find us. And this is, I always said it was the first event we were hosting this year, but there are a lot of news upcoming. So if you follow us, you'll be notified of our events. And that's it. It was a pleasure to be here. And that's all I've got to say.
That's it, guys. Thank you very much. I hope to see you, everyone, in Rio de Janeiro in a few moments after the vaccines. Rosie?
No, I just want Gabriel to ask René, Valéria, Dennis. Dennis is here. I don't know, to come to the room and say something. René and Valéria, some words, please.
René?
Valeria, I mean, I just have to reinforce the appreciation, my appreciation words for all the effort put on this event by, I mean, you guys that have hosted the event and have been with us effortlessly and for many days discussing and writing schedules and scripts, and it was really really good. And for all the scientists that have contributed with amazing and interesting elements to put together this great event, truly multidisciplinary event. and it's been such a pleasure and such a delight to see you all shine on our screen. So thank you very much.
Please turn on the microphone.
Yeah, the microphone's off. Maybe
Sorry.
Gabriel can. Yeah.
Aqui. I'd like to say that it was a great pleasure to be here with you. I think that it was a great opportunity to learn, to discuss several aspects of insect galls, and I am really waiting for the next symposium.
Maybe Marcelo now. Marcelo, would you like to say a few words?
Hello, everyone. It was an honor to participate in this amazing event. I learned a lot, and thanks to the Anatomical group for this event, especially Bruno and Rosie for hard work for this organization. Congratulations for this brilliant event. That's it. Thanks for all.
Okay, everyone. Thank you all for inviting us for this, for letting us host this event. It was, again, challenging, but really, I forgot the word, sorry.
I was muted. I was saying follow us on social media and see you soon. Bye-bye. Bye-bye.
Bye-bye.